Hi, John. How are you doing?
Hey, good. How are you, Dr. Lin?
Great. Great. Looking forward to it.
Yeah, this should be good. I'm looking forward to it. We got a lot of registrations.
We have just a few more minutes. We're gonna give everyone time to join and get started in about 3 minutes. Welcome, everyone. I think we'll just give it 1 or 2 more minutes to it looks like we're having a slew of people joining right now, so just bear with us and we'll get started momentarily. Alrighty, well, I think we'll go ahead and get started. So on behalf of Biocytogen, I'd like to welcome you all here to today's webinar. My name is Jenna Frame. I work in the marketing department here at Biocytogen Boston. We're very excited to continue this new webinar series that we've started during the pandemic in order to provide more resources to our community. We hope that these series will be educational, offer a deep dive into models and services that we offer here, so you can help learn more about the services that we're offering and how we can help to support you. So before we get started, I'll just quickly introduce our speaker and panelist who will provide their expertise today. So our speaker is Dr. John Charpentier, Business Development Manager at Biocytogen Boston. So John has his PhD in immunology. He previously worked on T-cell activation at Tufts and got his PhD from University of Michigan, where he studied macropinocytosis and T-cells. And as a business development manager here at Biocytogen, John provides comprehensive support for new and current clients. And then Dr. Ching-Kong Lin, who is joining us today for the Q&A session, has extensive research expertise in the field of molecular genetics, has held leadership positions at Wyeth, Pfizer, and served as the director of the Laboratory for Genetic Engineering at the Center for Genetics and Genomics Research at Harvard Medical School. In 2018, Dr. Lin joined Biocytogen as Senior VP and currently serves as CEO of Biocytogen Boston. So thank you both for joining us here today. And now, without further delay, I'd like to invite John to share his presentation with you. And we will have some full questions throughout and bring your questions for the Q&A session afterward. Thank you.
Thank you, Gina.
Yeah, thank you, Dr. Lin, for joining, and thank you, Jenna, and all of you for attending. So my name is, as Jenna said, is John Charpentier. I'm a business development manager here at Biocytogen Boston. I'm really excited to speak with you today about our humanized CD3 models. First, I want to give just a little bit of introduction and background to our company and the products and services that we provide here. So Biocytogen was founded in 2008 in Worcester, Massachusetts. Our initial focus was on genetic engineering, having developed proprietary methods to improve the rates of homology-directed repair and knock-in efficiency. But since then, we've generated over 3,000 custom animal models for academic and industrial clients worldwide. But we've leveraged the same technologies to create an array of off-the-shelf humanized rodent and cell models to support the research activities of our clients. So now we're one of the largest animal model suppliers in all of China. We opened our branch in Wakefield, Massachusetts, just north of Boston in 2018, with the goal of expanding the range of our capabilities as a contract research organization. So one of the biggest areas of recent focus is our antibody discovery platform. So we leverage our advanced chromosome engineering technology to develop the RenMab mouse. That's one thing I really want you to leave with today is to go and look into the background of the RenMab mouse. Now, this is a mouse that contains an entire immunoglobulin variant domain, the human sequence, including the surrounding non-coding regulatory regions. So it's replaced in situ and generates fully human antibodies. They're a really powerful platform for therapeutic antibody discovery. And what's more, after identifying antibody hits for your target of interest, we offer additional models and tools to facilitate the later stages of antibody drug discovery, including humanized mice for immuno-oncology target testing, cytokine and cytokine receptor humanized mice, as well as immunodeficient mice that are suitable for human immune system reconstitution. So our goal is to be the one-stop shop that assists you from target discovery all the way through to IND application. And so here's a brief outline of what I'm going to talk about today. I'll discuss first generally the challenges of translating basic science research discoveries to clinical application and how improved models can help bridge the gap. Next, I'll provide some case studies and context of how our humanized CD3 models are generated and validated. And then lastly, I'll discuss how these models can be integrated into antibody discovery platforms. And with the time that's remaining, we'd like to take your questions about any of these topics. So part one, challenges in clinical translation. I like to think of this as sort of, why do we need humanized mouse models? As all of you are doubtless familiar with, therapeutic candidates that are developed to capitalize on basic science research discoveries, most of them fail to find clinical application for any of a host of reasons. To give just one example, antibody therapeutics that perform well in preclinical studies often fail in clinical trials because of poor cross-species reactivity. So one proven way to reduce therapeutic attrition and to improve the odds of translation success is to refine the methods and the models that are used to evaluate the safety and utility of new drugs and biologics. And so how do we do this? There are a number of approaches one can take. As I alluded to at the top, here at Biocytogen, we've invested a great deal of our resources in improving preclinical rodent models. In particular, this means replacing murine genes with their human homologs, resulting in the expression of human transgenes in the mouse model. By strategic development and use of these models, which recapitulate human immunity either in part or in some ways in whole, we aim to accelerate the pace of preclinical discovery and development and significantly narrow, if not close, the translation gap. So at Biocytogen, we have a range of humanized animal and cell models. And for the purposes of this talk, I'm going to focus really on the humanized CD3 mice. But there are many other tools available for those of you that are interested in developing therapies that modulate immune responses, including all of the varieties that I just listed and are shown here. So these models have all been very widely employed and successful in early preclinical studies, tox studies, safety studies, as well as more advanced efficacy studies for example, immuno-oncology and autoimmune therapeutics. So our technical teams have worked very hard to generate and rigorously validate these humanized mice, some of which are singly humanized, others multiply humanized. There's just a few examples listed here, including the CD3 models that we have available. But for a more comprehensive list, you can check out our model catalog or visit our website to search for just your model of choice. So what about the utility of humanized mice, and in what ways can they help us close the translation gap? So some of the advantages of evaluating drug efficacy in these models is that the animal has an intact immune system. So as opposed to immunodeficient models that are engrafted with human PBMCs or CD34 positive HSCs, which might not engraft and differentiate and proliferate properly. In vivo studies in these mice enable mechanistic insight into pathophysiology and therapeutic mechanism of action at much lower cost with higher numbers of animals and greater ease than similar studies would be in non-rodent models. The primary disadvantage, of course, is that the mouse and the human immune system are not entirely identical, as we know. So in some cases, the disease setting and the therapeutic activity in humans may not be completely or accurately recapitulated. To ensure the preservation of in vivo signaling interactions and function, one often needs to humanize both receptor and ligand pairs, and this can add substantially to the cost of the model. But on the other hand, these models uniquely allow for the testing of bispecific and combination therapies. So now I'm going to walk you through just a couple of case studies focusing, as I said, on our CD3E mice to give you a better sense of how we generate them and validate them. So CD3 is obviously a popular development target because of the central role that the complex plays along with the TCR in mediating T cell activation and downstream functions. So for example, bispecific T cell engagers that are targeting the CD3 extracellular domain with one paratope and a tumor-associated antigen, for example, with a distinct second paratope. Bispecifics like this have become a very hot area of development. And so the design facilitates a cancer treatment strategy that's depicted on the slide here, wherein the antibody cross-links CD3E on a cytotoxic T cell and a tumor antigen on a tumor cell to promote inflammatory cytokine release and anti-tumor activity. So a number of anti-CD3 drug candidates have been developed and successfully marketed, but there are still significant challenges in identifying those that have the best safety and efficacy profiles to advance to human trials. So this is in large part because antibodies that specifically activate murine CD3 cannot effectively activate human T cells and vice versa. And this is primarily due to the low sequence homology between murine and human CD3 chains, which you see here ranges from about 50% to 60% from human to mouse. So humanized CD3 models are very much needed for the testing of novel CD3 therapeutics in vivo. So in the next few slides, I'm going to highlight the design and characterization studies we performed for our humanized CD3 epsilon chain model to ensure normal T cell functionality. And so we hope that this will also give you a sense for how generally we design and validate our models here at Biocytogen. In this case, the extracellular domain of human CD3E, which is shown here in pink, was inserted in situ in the murine CD3 epsilon locus, completely replacing the murine mirroring extracellular domain. And so if you then evaluate CD3 epsilon human mRNA expression by RT-PCR, we can see that the murine protein is only detected in the wild type mouse, and human CD3 epsilon is only detected in the homozygous CD3E humanized mouse as expected, and that's outlined in the little pink box there. And then importantly, when we measure expression of CD3 by flow cytometry, we detect only human CD3 on the surface of T cells of the humanized CD3E mouse, and only murine CD3 on the surface of T cells from the wild type mouse, as you see on the top left. So these are findings that are consistent with one another, and suggestive of reasonable expression of the transgene. So previous attempts to generate CD3 mice, humanized CD3 mice, I should say, have met major challenges. Early attempts have resulted in completely defective T cells, or other models that show impaired or disabled T cell responses. In our model, gross assessment of lymphoid organs from these mice does reveal reduced thymic mass, presumably due to hypoplasia of thymocytes, but spleens are normal weight. So studies with other humanized CD3E strains have suggested that the extent of thymocyte depletion that we see in these CD3 epsilon models is correlated with transgene copy number per cell. So this is going to lead to, I think I got the question, my poll questions in the wrong order here, but let's do the first poll question first because I skipped past it. Let's bring that one up. And that poll question relates to what was the first CD3 therapeutic that was marketed? So Stone, if we could pull that first poll question up. There we go. So I guess we'll give that about 30 seconds for you to vote on it. The first CD3 specific monoclonal. So by way of background, this monoclonal was developed in the late 70s, was approved in 1986 to prevent allograft rejection for kidney and heart transplants. We'll give it just a few more seconds, I suppose. All right, I'm going to stop it now. So yes, the correct answer is C, muromonab, which is also known as orthoclone OKT3, was developed in the late 70s, and was the first monoclonal that was approved for any indication at all. So it was the very first monoclonal, and now monoclonals are a $150 to $200 billion global market. So CD3 has played quite a big role in the generation of that. So getting back to the characterization of the CD3 epsilon mice, I see here now, this is where my second poll question could be. So we'll go ahead and get that over with too, because it's relevant here. If we could bring up question number 2. So this is a question related to the thymocyte depletion that we see in these models. So thymocyte and consequently peripheral T cell depletion can result from which of the following expression of CD3? Overexpression of CD3E leads to thymocyte depletion, underexpression, or both over and underexpression. And we'll go ahead and just give that about 30 seconds. The important take-home point here being that expression of CD3 epsilon is carefully regulated and critical for the development of thymocytes and T cells. All right, I'm going to go ahead and stop it. And it looks like, yeah, most of you 44% got it right. Both over and under expression of CD3 epsilon is associated with thymocyte depletion. So it's a very tricky technical problem to get around to modulate expression appropriately and consistent with normal thymocyte development. And that's because precisely controlled CD3 epsilon expression is critical for TCR rearrangements that happen in the double negative stage. So before progression to the double positive thymocyte stage, CD3 expression is important for survival signaling. All right, great. So I'm going to go ahead here. And then, so despite the observed thymic hypoplasia that we see, humanized CD3E mice exhibit normal immune cell profiles when compared to wild type C57 black 6s. So here we can see by flow profiling comparable numbers of lymphocytes, of NK cells, myeloid cells, and that's in both the spleen and the thymus of the CD3 epsilon humanized mice. We also, though, want to compare relative numbers of cells of different T cell subsets. So here we see, similarly by flow profiling, comparable frequencies of CD4s and CD8s, as well as FOXP3 positive Tregs. And that's true in the wild type and humanized CD3 mice in both the spleen and the thymus. So despite the underweight thymus, we do see normal proportions of distinct cell subsets. And so one measure we're most interested in ensuring the preservation of these humanized CD3E mice is specific T cell activation. So to examine this, we isolated T cells from splenocytes in both the wild type and the CD3E humanized mouse. We stimulate them in vitro with anti-CD3 and 28 and let them sit for about 48 hours. And we see that while wild type mice upregulate activation markers like the high affinity IL-2R alpha chain or CD25 and CD69 in response to anti-mouse CD3, the humanized mice only activate in response to anti-human CD3 epsilon antibody and co-stimulation, not the murine CD3 epsilon antibody. And so that's consistent with our expectations as well. Another measure of preserved functionality is proliferation of T cells following appropriate stimulation. So to compare the proliferative responses in wild type and humanized mice, we performed in vitro CFSE dye dilution time course experiments over 72 hours. And these confirm that stimulation of CD4 and CD8 T cells from each induces proliferation very strongly, as indicated by the leftward march of the dilution peaks here on the bottom right. And indeed, the proliferation of these cells was comparable to that seen in stimulated T cells over 72 hours in wild type mice, as you see there. So naturally, we're also interested in ensuring that the T cells from our humanized mice also secrete the expected cytokines in response to CD3 stimulation. And so comparing in vitro cytokine release between splenic T cells and wild type and CD3E humanized mice, we see that when the latter are stimulated with anti-human CD3 antibodies, and that's the white bars that you're seeing in these graphs, they release comparable quantities of interferon gamma and IL-2 with similar kinetics to wild type over 72 hours. So this is all a consistent picture of appropriate subsets at the right frequency, of appropriate activation and appropriate proliferation and cytokine release compared to wild type. But of course, we also want to ensure that the collaboration between T and B cells is not perturbed. And so to examine humoral responses in these mice, we immunize them and a wild type cohort with ovalbumin. And as shown in the left two panels here, both wild type and CD3E humanized mice produce comparable titers of ovospecific antibodies after a second and third round of immunization. And so this suggests that humanization of CD3E does not impair productive T-B cell collaboration. And even more encouragingly, we see on the right there that the antibody isotype distribution in humanized mice is comparable to that seen in wild type as well. And then finally, really getting at the central question we're most interested in when employing these models for development purposes, to assess whether T cells from humanized CD3 epsilon mice can efficiently eliminate tumor cells in vitro. We perform cytotoxicity assays by pairing splenocytes from our humanized mice and murine MC38 colorectal tumor cells that are also expressing PD-L1 to recapitulate an immunosuppressive tumor microenvironment. So the cells here were mixed at the indicated ratios and then incubated with a bispecific antibody that targets both PD-L1 and human CD3. And as the graph shows, anti-tumor activity was evident at both cell concentrations, but was enhanced with increasing concentrations of the bispecific. And so taken together, these data offer really compelling evidence that human CD3 is functional in these mice, at least according to in vitro assays. But what about in vivo studies? So far I've shown you a bunch of in vitro stuff that suggests that the function of these T cells is preserved and they develop and differentiate normally. But next I want to show you some data from our in vivo validation studies. So in the first case, humanized CD3 epsilon mice were inoculated with murine MC38 tumor cells again, and then were treated with either an IgG control, which is shown in the blue line here, or an anti-human CD3 antibody that induces T cell depletion. And this is critical for interpreting the experiment. This antibody induces depletion of T cells by activation-induced cell death. So as shown on the graph, mice that are treated with higher doses of the antibody, the 3 mg per kg dose in the red-orange line, they experience more rapid tumor growth than those receiving lower doses or controls, which is the expected result. The significant depletion of peripheral T cells in this case was associated with a relative enhancement of B cells, which is shown over here on the right side, in both of the right panels. Overall, though, the study confirms that humanized CD3E mice are a powerful tool for in vivo efficacy studies that are evaluating anti-human CD3 therapeutics. But what if we want to look at the ability to promote tumor killing of our engineered mice, as opposed to activation-induced cell death? So in the studies that we summarize here, our team performed a series of bispecific antibody efficacy assessments in the humanized CD3 epsilon mouse model. And in the first panel here, we see an MC38 tumor cell line that's engineered to express now the B cell marker CD19. And so these were MC38 cells were inoculated into the humanized mice. The tumor volume was then followed over several weeks in cohorts that were treated variously with differential doses of blinatumomab, or Blincyto as the trade name goes, which is a bispecific that's approved for treating acute lymphoblastic leukemia that recognizes human CD19 on the one side and human CD3 on the other variable domain. And we see that higher doses of the antibody here are correlated with proportional reductions in tumor volume over time compared with vehicle controls. So that's as expected. But similar results were obtained in a study of human CD3E mice that were inoculated with MC38 again, but treated with a bispecific antibody to human CD3 and an undisclosed target antigen that was discovered by a collaborator. But again, the important thing to notice here is the trend that higher doses of the bispecific antibody were correlated with decreased tumor burden. So then in the third study, that I'm going to show you, humanized CD3E mice bearing MC38 tumors expressing the tumor marker EpCAM were treated with a bispecific that targets both human CD3 and EpCAM. And once again, we confirmed that higher doses of human CD3 specific bispecific antibodies were associated with a reduction in tumor volume compared to the lower dose cohorts and controls. And so altogether, these studies demonstrate the value of the humanized CD3 epsilon model for testing the efficacy of bispecific antibody candidates. So now very quickly, I want to just tell you a little bit about one of our newer models, the humanized CD3 epsilon delta gamma mouse, or EDG mouse. The goal here was to more accurately recapitulate human T cell biology in the mouse. And so our team has recently turned to generating and characterizing this second generation model in which the entire CD3 complex is humanized. So in this model, the entire coding sequence of the CD3 chain, the epsilon, delta, and gamma, as well as some of the human untranslated region, were inserted in situ in the corresponding murine locus, or loci, I should say, it's more than one, and replacing the murine genes. So again, we perform RT-PCR and blots to confirm mRNA expression of each chain, and you see that outlined in the pink boxes here. And then importantly, expression of human CD3E is detected on the surface of the heterozygous humanized EDG T cells by flow cytometry as well. So all of this looks consistent. Gross examination of lymphoid tissues from the humanized mouse didn't reveal any obvious abnormalities. There was no significant difference in spleen weight or the number of wild type, or cell number, I'm sorry, between the wild type and the humanized mice. However, thymic weight in the humanized CD3 EDG mice was still slightly lower than that of the wild type mouse. And this relates to the problem that we discussed before about expression during thymocyte development of CD3E. But But the total number of thymocytes in the EDG mice, again, were very slightly lower than the numbers harvested from wild type mice, as shown on the bottom right. So this is an area of concern, but we would first want to verify that there is normal proliferation, differentiation, and other responses to address any or alleviate any concerns about the slight reduction in thymic weight. And so importantly, as we showed with the CD3 epsilon model, the stimulated T cells from the EDG mice appropriately proliferate in response to specific stimulation with anti-human CD3 antibody. And that's shown here in the dye dilution peaks in this proliferation assay. On the bottom right, we see that the proliferation response in the triple humanized or triple chain humanized mice are similar to the wild type. Similarly, as with the CD3 epsilon model, stimulated T cells from the humanized EDG mice also secrete cytokines normally in response to anti-human CD3 and murine CD28 co-stimulation. And that's shown here by the orange bars. So secretion of interferon gamma and IL-2 72 hours post-stimulation was similar in these mice to their wild type counterparts that you see outlined in red there. That were stimulated with CD3 and CD28, I'm sorry, outlined in purple. So, and then to ensure that the EDG humanized mice exhibit normal humoral responses, we again performed ovalbumin immunization studies. And you see that in the left panels, both the wild type and humanized mice are observed to produce normal levels of anti-ova antibodies after 3 rounds of immunization. And again, these antibodies exhibit a normal antibody isotype distribution, suggesting that in these mice, they have normal cellular and humoral immune responses. We've only recently, however, begun in vivo efficacy studies with our new CD3 EDG model. But I'd like to tell you a little bit about what we've learned so far from that. In the initial testing, we again performed T cell antibody depletion studies to determine whether blockade of anti-human CD3 and depletion of T cells would enhance tumor growth consistent with expectations. And so as before, on the left side here, you see, oh gosh, I think the image isn't loading. On the left hand here is I'm sorry about that, is supposed to be a graph similar to the one that you previously saw that shows that higher doses of human CD3 blocking antibodies do in fact enhance tumor growth. What I can show you, and looks like it did load here on the right side, is confirmation that the therapy depleted T cells but not B cells, as shown by the green bars in both the peripheral blood and in the tumor microenvironment. So I apologize for that image problem. We'll get that fixed. That information is available on our website, or if you contact me, I'm happy to share it with you. But we look forward to sharing our new data as we continue to learn more about the potential of the CD3 EDG model in vivo efficacy testing of bispecific antibodies. We're pretty excited about it. So the models that I've described today are just a few of the many, as I said, that we've generated here at Biocytogen. We're generating more all of the time. I encourage you to head over to the website. You can download our full catalog there or contact any one of us by chat or email, and we're happy to send that to you. You can also, for any of these targets that you might be interested in, you can search on the top right in the search bar on our website, and it'll bring up any information we have about the target of your interest, including things like targeting strategy and some validation details. So happy to show you those. Another thing that I want to tell you about that's a really important tool in these studies for testing novel immunotherapies especially are the off-the-shelf humanized murine MC38 colorectal lines expressing a wide range of human immune targets. So in addition to all of these off-the-shelf lines and model generation, at Biocytogen, we're also happy to utilize our skilled gene targeting team, and they can generate custom humanized target cell lines for you. Okay, so to summarize what we've discussed today, we first talked a little bit about the challenges of translating basic science research discoveries into viable clinical entities. We described how Biocytogen validates and characterizes our off-the-shelf humanized CD3 models, looking at protein expression, immune cell populations, and functional assays using reference antibodies. And I hope I've convinced you that our humanized mice are really powerful models for robust preclinical testing and novel CD3 targeting therapeutics, including, mono and bispecific, multi-specific antibodies, but also combination therapies and preliminary toxicity assessments. And so our hope is that when you get to that stage of needing to do early pharmacology assessments after identifying your target and developing an antibody with us, we also have a very talented in vivo pharmacology team to assist you with those studies as well. Okay, so then now I guess we're gonna open it up to your questions. Hopefully you have a lot of them, and I'm going to invite Dr. Lin to join us as well. And thank you for your attention for my talk. Thank you.
Thank you, John. That was super informative. Okay, one moment.
So there's one question asking about the CD3 and other tumor targets. We have couples of the double humanized mouse model based on the CD3E, but we don't have that comprehensive every target. So please talk to us, our team, and John and our team. If you need that one, we can cross and to make a double for you easily.
Yes, I think due to our extensive gene editing expertise, anything is possible. So if you don't see the model you're looking for, just reach out. We might be developing it, and it might already be in the works, or we can, customize it for you. Let's see. So there have been several questions in the chat. Let me just go to the first question. So which would be better, using humanized murine tumor cell lines or human tumor cells engineered for a specific target?
Yeah, we want to take that question.
So I think
so it's a good question, Gina. Let me take it.
Sure.
So most of the case using humanized murine tumor model is very good. You can do very high throughput. But if your antibody is against of the human target and you prefer to use human tumor cell line, then means you need to use xenograft mouse model. Exactly. And then there is immunodeficiency mouse model. So it's two, both have pro and con. For fully mouse one, you can do the small throughput, but you use human mouse genetic background. But then you use the immunodeficient mouse and PBMC or CD34 regraft of the human immune system, then you can use the human tumor cell line. So they have pro and con.
Yeah, and it can depend on your target as well. So, our pharmacology experts will kind of work with you to help you design your study, based on the type of target that you're working with as well. Thank you, Dr. Lin. So how long will it take to generate a human target knock-in MC38 cell line, and that approximate cost?
So usually it will take 3 to 4 months, but sometimes it depends on your target. Some target is very popular and easily, when some target could be very challenging. And particularly for some target, if you knock out in the cell line, it could be lethal, then it's a very different story. So I think our team definitely can work with you to address your question. In general, you can say you can take it as 3 to 4 months, and the cost, again, that's like what I say, in the ballpark, 5,000 to 15,000, depends on your target.
Thank you, Dr. Lin.
I would also just jump in to add that that's right, that one of the advantages of working with our custom model generation team is that they'll also provide you with a number of targeting strategies. So there will be alternatives to work with if you run into technical challenges.
So I'm just going to go through the chat here because I think there are some questions. So we had several questions related to whether we offer CD3 tumor target double transgenics. And yes, we do offer several, including some multigenic humanized mice with our new CD3 EDG model. So be sure to check out our website. There was a question about SERP alpha mice and CD47. And yes, we have several combination models, double, triple models with humanized SERPA and CD47, including immunodeficient mice. And then, okay, there's a specific question. Do you have PK data from the bispecific T-cell engager efficacy studies? John or Ching Kong, do you want to comment on that?
I don't know specifically if we have PK data for that, but I'm happy to contact the team that conducted it.
The answer is currently we don't, but most of people concentrate on the efficacy and we don't look into the PK. But it definitely is a good question and it's doable.
Right. It definitely is a service that we offer. Have you performed infection studies with these mice?
So the question is, what exactly infection means? Do the challenging or what kinds of the infection you are mentioning? So it's a little bit tricky, the question. I don't fully get it. But in terms of these mice, they are behaving like wild type. So you can see in the wild type response, you can see in our CDE humanized mouse model or CD3 EDG model. So in fact, we do the immunization and then the titer is very similar or comparable to the wild type. We do the challenging. If that is the, you mentioned about the infection.
Right, and I would assume also that they're housed normally. They don't require any sort of antibiotics or anything like that. So, but we definitely look into it to see exactly what he means about infection.
Okay. Yeah, and I also would add that I have no doubt that, out of the thousands of clients that we've served with custom gene-targeted models, that they have then used them in infection studies in their research, just based on the sheer number of models we've generated.
So there was a question, have you shown that a TCE causes toxicity in a double transgenic mouse? For example, can you replicate on-target off-tumor toxicity?
Yeah, very, very challenging and good question. and when we talk about CD3 or CD3 we always face that questions. So the honest short answer is we don't look into so close, but we look at toxicity in terms of they read out. So currently we don't do solo study on this. So no exact answer for you. Sorry about that, but we definitely look into it.
Right.
Because cytokine storm and so on so far, it happened for the CD3 base all the time. So tumor toxicity and off-target toxicity need to be investigated for sure.
Okay. Oh, I think there was one question. Do the CD3 EDG mice have an advantage over the CD3E mice?
Yeah, I think they do in that the CD3 complex, is distinct heterodimers that function together with the TCR. So the extent to which all of those chains are humanized, I think, creates a more human T cell in the mouse model. So signaling functions, many of the conserved signaling motifs that are intracellular, the ITAMs and things like that, are going to be the same in mice and in human. But the extent to which the entire complex is humanized, I think it stands to reason would lead to a more accurate picture of human biology.
In addition to what John just mentioned, the difference between CDE and CD3 EDG is CD3E, we only humanize the CDE's extracellular domain. However, in the CD3 EDG, we humanize entire 3 chains, including extracellular domain and intracellular domain. Therefore, big advantage using for the 3 EDG, not only downstream signaling, but more important is if your CD3E or antibody bind with the complex instead of only 3E chain only, then 3EDG will have the advantage. So it depends on your antibody epitope, only on the CD epsilon or on the CD3EDG complex. I think that is the key difference.
Yeah, thanks, Dr. Lin. That's a great point. And just to reemphasize what he said, you know, the landscape of epitopes that are available to you for development when targeting any part of the CD3 complex is just going to be much bigger than the CD3 epsilon chain alone.
Okay, thank you. I think we have another question coming through in the chat. Have you compared the binding to CD3 from your EDG transgenic mice to binding to CD3 on human cells? How is the binding of antibody clones such as OKT3 that bind to a conformational epitope?
I presume that's a question about binding affinity. It's what it sounds like. I don't know that that's been measured directly, and it would be interesting to know. But I really think the important part here is the functional readout. So we know that the cells are getting activated. We know they're responding normally. So it must be binding with comparable affinity. But it is a good question. I would imagine it's within the same range of magnitude.
And for a pharmacology study, would you cross-compare OKT3 with your novel antibody?
Yeah, I think so. I think that's exactly the sort of thing that you would do.
Okay. Thank you, everyone, for those questions. Let's see. All right. So as we wait for any final questions to come through, I just want to advertise that our next webinar is going to be December 1st, I believe at noon. And the topic of that webinar is going to be focused on our humanized cytokine mice, which can, be powerful tools if you're interested in modulating inflammatory disease. So we have several new models for, humanized IL-4 and other cytokines, and you're welcome to check those out on our website. In the meantime, if you would like to join our mailing list, if you haven't already, to make sure that you don't miss any important emails and promotions and registrations for the upcoming webinars, please make sure that you're on our email list. And contact us at info@biocytogen.com if you have any other questions about, the CD3 models in particular, or any other models or services that you might be interested in. It looks like we have a couple more questions, so we'll just quickly address those. Let's see. Will the human CD3E mouse generate antibodies against the CD3E extracellular domain, or ECD?
The answer is no.
No, okay. And did you compare anti-PD-1 tumor efficacy in the humanized CD3E knock-in versus wild type?
So we did. When we do the CD3E knock-in model, we use the anti-mouse PD-1 as a benchmark. So we do see the efficacy over there. So, but regarding compare direct with wild type, I don't think we have tighter control over there. It's not apple to apple, but we do use the mouse PD-1 antibody in CD3E humanized mouse model efficacy study. We see very good efficacy. Okay. So it should be the comparable, but we may not have the direct comparison data in the same study. So we should look into it. Thank you. Very good question.
Thank you. Okay. Well, thank you all so much for joining our webinar today. And thank you, John, for presenting, and Dr. Lin for joining us for the informative Q&A session. And we hope to see you next month.
And thank you, Gina, for the hosting this event. And I see many of the old friends and new friends. And thank you for joining us and looking in for our future webinar. You are welcome to join. And we have a lot of model than what you expected. So, and in case you don't see the model you are looking for, talk to our team. We can generate both cell line or mouse model for you, for sure. Thank you.
Thank you.
Thank you both, and thank you all for attending.
Okay, have a great day.
Good job. Thank you.